Good afternoon, everyone. My name is Dr. Saranya Chumsri, and my colleague is Dr. Brenda Ernst. So we're both from Mayo Clinic. So we would like to welcome you to our career development session on patient-driven protocol. You know, I think in the modern era of medicine, it is very imperative for us researchers and also clinicians to incorporate these patients' voice, patients' insight into our protocol development, because it's not going to only make the protocol more effective and easier to enroll, but also make it much more relevant for patients. So today we have a couple of esteemed speakers today from patient advocate, and we have people from academia, industry, and also FDA to help enlighten us on how we can, effectively integrate these patients' voice and insight into our protocol development to make it meaningful. We will start the session with a couple questions on Slido, just to know where you are, who you are in the audience. So we'll start with, so if you can bring up your phone and scan this QR code, please. Okay. So we see the phone is down. Okay, we'll start with the first question. So please select who are you, whether you are a patient advocate, clinical researcher, basic researcher, clinician engagement in research, or others. Please choose your, and you can, if you missed the first QR code, you can also use this QR code too. Okay, looks like a lot of us are clinician engaged in research, so that's great. So we can get you to do more patient engagement research. Okay. So we good? So let's go to the next one. So where are you in your career, whether you are a resident, fellow, or early career, within 1 to 5 years, or mid-career, 6 to 15, or late career? Please enter your response. Oh, okay. Yeah, we have a bit of mid-career. Okay. So next one is, are you familiar with patient-driven protocol? So first one is very familiar, somewhat have some knowledge, or not familiar at all. Please enter. Okay, this is great. So then hopefully you'll get something out of our session today. And then the last question, have you written a clinical research protocol? None, or a couple, 4 to 6, or more than 6? Please enter your answer. Okay, so we do have the mix of between none and a couple. Okay. Thank you. So I think now we're going to start off our session with Debbie Denardi. So she's our advocate, and we really want you to hear from patients' perspective first on why is it important for us as a clinician and researcher to incorporate their insights and voice into our protocol. Thank you.
Good afternoon. And can we toggle the mic to the table? It's good. Yes. Good afternoon, and thank you for being here. My name is Debbie Denardi. I'm originally from Argentina. I grew up in a family with a lot of breast cancer, ovarian cancer, and also colon cancer. So I knew that at some point in my life I was going to be diagnosed. And in 2010, I was diagnosed with triple-negative breast cancer. And back then, I went to 4 different oncologists. The first 3, they did not recommend genetic testing. Even though I had a lot of family history, the 4th one recommended genetic testing. And it came back that I had a BRCA1 mutation, and that, my sister too, and that saved her life because she was able to do all the prevention and the surgeries related to the BRCA1 mutation diagnosis. Moving forward to, well, I did all my treatments, my surgeries, etc., and I finished with that. And I continue learning about different opportunities for patients to participate in research. And this where last year in December, I decided to participate in a clinical study that they were recruiting patients that had the BRCA1 mutation, and they wanted to develop a biomarker to detect pancreatic cancer earlier. So I decided I'm going to enroll to this study. When I went, I talked with the gastroenterologist, and he said, well, with your family history from your dad's side, you should get tested again. And this is where I was testing, and I also carry a Lynch syndrome mutation. And for me, that is really a very important piece of information, because now my sons, they were tested, and they will, both of them, they carry the Lynch syndrome mutation. They will be able to do the screening early enough before they would develop colon cancer. So at the age of 30, they are going to start with their colonoscopies and everything. So all that information, it's really important for us patients, but also in order for us to participate, we need to be educated. We need to receive information. We need to be able to have the opportunity to participate in clinical research. And also, as you can realize, I'm Latina, and I really advocate a lot for Latino participation in clinical trials. So we'll talk a little more about that, but I wanted just briefly to give you that experience that really, as survivorship is so long, we need to continue screening patients. Thank you.
Thank you very much, Debbie. And a little bit for house thing. So we're gonna keep the Q&A at the end of the session. So we'll have the speaker talk first. So we'll start off with Dr. Tatiana Prowell. So she's from the FDA and also Johns Hopkins. So she'll start us off with how to design patient-centric clinical trials. Dr. Prowell.
Hi, thank you so much for the invitation to be here, and to all of you in the room for joining us. I'm going to be speaking today about designing patient-centric clinical trials. Let's see, oops. All right, there we go. I have nothing to disclose. Sorry, I don't know why this is happening. Okay, great, thanks. All right, great. So we'll just begin with an outline here. I'll speak just for one slide about recent drug approvals, and then we'll spend most of our time talking about three major things that I think would make trials more patient-centric: modernizing eligibility criteria, trials with decentralized elements, and advocacy-informed trial design. And then finally, I'll just close with two slides to make sure everyone in the room is aware of two tools regarding compassionate use for patients who may not qualify for a trial. Okay, so this figure is actually from my colleague at FDA, Shaylee Arora. She did a lovely overview of a decade period of oncology drug approvals from 2010 to 2020. In that time, we approved 30 new drugs for breast cancer. I'm delighted to say that this slide is both difficult to read and totally out of date, not a thing that we normally celebrate, but in this case, it's wonderful because it really reflects the number of drugs that have been approved and the continued pace of approval beyond 2020. So since this slide was made, we've had approval of adjuvant immunotherapy in breast cancer, adjuvant PARP inhibitors, adjuvant CDK inhibitors, approval of a first drug to target ESR1 mutated breast cancers, and more. And so this is obviously a very exciting time in breast cancer and very good news for patients. So the first topic I'd like to cover is modernizing eligibility criteria. So why do we consider eligibility criteria at all? Why do we even have them in clinical trials? We rely on these really for three key purposes. The first, and probably the most important, is to protect the safety of clinical trial participants. But we're also importantly looking to define the population so that we know to whom these results will apply on the back end, and also to maximize our confidence that the results of the study that we see, both in terms of safety and efficacy, are actually attributable to the study drug and not, for example, another condition or the natural history of that disease. But concerns for patient safety and concerns for being able to define the population have led us to exclude a lot of patients that will be represented in the post-approval population and will receive the drug. And so these include a variety of people, but including patients with brain metastases or leptomeningeal disease, patients with a marginal performance status or many common comorbidities, patients with organ dysfunction or limited marrow reserve, both of which are common, particularly in patients later in disease or patients who've had a lot of prior therapy. People with HIV or hepatitis B or C infections, people with extremes of age, both directions, young and old, and patients with prior malignancies of other types. And then finally, we've also seen exclusions of patients who, for example, have or have not been treated with a certain class of therapy. And the result of all of these exclusions, which I think it's important to say, all came about with very good intentions. The result of all of this is slow accrual to trials that are less generalizable to the population that will receive the drug post-approval. So that recognition led to a working group being formed by American Society of Clinical Oncology, as well as a patient advocacy group, Friends of Cancer Research, and led to an entire special issue of the Journal of Clinical Oncology in 2017. And then a subsequent later second issue of Clinical Cancer Research that was dedicated to this issue of modernizing eligibility criteria. And there were papers, I would encourage you to look at both of these. There were papers that were issued pertaining to inclusion of adolescents, for example, in clinical trials, inclusion of older adults, inclusion of people with all the things that I said from the previous slide. And in the case of breast cancer, including patients of any sex or gender unless there's a compelling scientific rationale to exclude them. And following all of these publications, NCI updated its standardized template that's used for all NCI-sponsored trials to be consistent with these recommendations. And so what's the impact of that? Well, there have been several papers that have looked at this, but here's just one example. This paper that was published by Harvey and colleagues in Clinical Cancer Research was a retrospective observational study using ASCO's Cancer Link. And they looked at patients with non-small cell lung cancer over a 7-year period from 2011 to 2018, 10,500 patients. They considered just 3 selected eligibility criteria: brain metastases, prior malignancy, and renal function. And what they found was that using traditional eligibility criteria, the things that many of you probably copy-pasted from your mentor, who copy-pasted it from their mentor, 48% of patients would have been excluded from these trials, and that was decreased to 2% using the modernized eligibility criteria. So really an extraordinary difference for patients interested in joining a trial. So of these working groups, I was very proud and honored to get to be part of the working group that was dedicated to brain metastases, because this is a problem that's, of course, very common in breast cancer, which is my own area of clinical practice. So just to give you a little bit of background about the magnitude of this problem, in the US, about 70,000 patients per year develop brain metastases. In patients who have HER2 positive or triple negative metastatic breast cancer, up to half of those patients will develop brain metastases prior to death. And historically, these patients have been excluded from all clinical trials in oncology. And exclusion of patients with untreated, so meaning people who've not had radiation yet, brain metastases remains really very common and I would argue the norm. And this group, as a result, has high unmet medical need. So why do we want to modernize eligibility criteria? I think it's important to just say very clearly, as a regulator standing up here before you, patients with brain metastases, including in some cases patients with untreated brain metastases, belong in clinical trials, period, the end. Why? This helps us improve patient access to investigational agents. It gives us as a community the opportunity to learn from the experience of all of these patients. From the standpoint of industry, this can inform a go, no-go decision in crowded development areas. So for example, rare biomarker-defined subsets or biomarker-defined diseases. And important to both clinicians and patients as well as drug developers, this can enable us to differentiate between drugs in a class, right? So you've got, a CDK inhibitor, you've got immunotherapy, you've got whatever class of drugs you're thinking about. Knowing that a drug does or does not have activity in patients with brain metastasis may be very important in your selection as a patient or as a clinician of which of those many drugs in that class you would choose to use. And again, from the standpoint of industry, for those of you who are in the room who are in industry, this can also enable you to have a novel indication or a labeling claim, and I'll show you an example of this in a moment. It can also serve as the basis for one or more expedited programs. So this would be things like a priority review clock that speed up the date by which we have to make a decision on approval. This could be the opportunity to get a breakthrough therapy designation earlier in development, which enables you to have very close, real-time almost access to the FDA as you encounter challenges in your development program and more. And finally, it may limit the need for post-marketing studies. If we know that there is a group that was underrepresented or unrepresented in your trial, and we think that they will be significantly represented in the post-market setting, we will often ask the company or compel the company to do a study in those patients in the post-marketing setting, so including these people in your initial registration study can avoid the need for that. And so I think most importantly, and this is what I've heard again and again from patients, people with brain metastases will be treated in the post-marketing setting, and they deserve, and clinicians need, information, enough data to weigh the risks and benefits of a drug in people like themselves, not extrapolating from a group that was substantially different. And I see several advocates in the room nodding their heads, so I hear you, and thank you for saying this. And so here's one example of this, the HER2 CLIMB study, which was in patients with HER2-positive metastatic breast cancer, randomized patients to receive capecitabine and trastuzumab with or without tucatinib, which is an oral HER2-directed therapy. And this study not only met its primary endpoint and improved progression-free and overall survival in the overall population, but when you look specifically in the subset of patients who had brain metastases at study entry, which was almost half of the study, you likewise see that same benefit demonstrated in those patients. You see intracranial responses. And this led to, if you see down at the bottom of the slide, an indication for the sponsor that specifically mentioned including patients with brain metastases. Why does that matter? Why would a company care about that? Isn't that widely known from the publication? Yes, but what's in the label is directly related to what a company can say in their advertisements and what they can say when they come talk to you in your clinic, right? And so they care deeply about what that indication says and what is in the label. So now I'd like to pivot to talk about trials with decentralized elements. So it's not news to anybody in this room that the pandemic disrupted almost everything, including oncology research to a great degree. And so this paper looked at an 8-year period, the same 8-year period of October to May, over 5 successive years, and just looked at the number of oncology clinical trial launches. And if you look at the bottom line down there in turquoise, that's 2019 to 2020, so that's really the pandemic period. And you see that really starting in late December or early January, when we had the very first inklings that something was amiss, and this was going to, of course, turn out to be the COVID-19 pandemic, you see this fall in clinical trial launches, and this is obviously very dramatic by the end. And then when we look at not just launches, but accrual to ongoing trials and impacts on oncology trial conduct, we see something very similar. So accrual to NCTN-sponsored trials fell steeply by more than a third in the first 3 months of the pandemic, and remained below normal for a prolonged period of time. And global enrollment to oncology clinical trials was likewise substantially impacted, with 20% of institutions in the US and Asia reporting no enrollment whatsoever, and 20 to 86%, depending on which continent you're looking at, of centers in different regions reporting that trial accrual had slowed. So this all led the international oncology community to say, how can we urgently reimagine clinical trials to ensure that they remain accessible to patients and to ensure that the data from ongoing trials remains interpretable, because the last thing we want is a patient's sacrifice of their own time, maybe a decision to enroll in this trial versus to do something in the standard of care, and then not be able to learn from their experience, or worst of all, not be able to appropriately ensure their safety. And so this led to a number of initiatives, and these things, to be clear, were already being discussed. Within the FDA, we already, prior to the pandemic, we had begun working on a draft guidance on decentralized clinical trials a few years prior. I think what happened was that COVID-19 forced us to say urgently, can we stand this up right now, like in the next couple of weeks? And so it's important to just step back for anyone not familiar with this term and say, what is a decentralized clinical trial? It's a clinical investigation in which some or all of the trial-related procedures and the data acquisition take place remote from the investigator. And so obviously this can encompass a variety of things. This could include remote consent, electronically consenting people. This could include doing visits via telehealth rather than having patients come in. It could include utilizing local healthcare providers, local laboratories, or even home blood draws or tests sent to patients to collect their own blood. It could include local imaging. And then in addition, the other things that I think we more conventionally think of with a decentralized trial, like delivering the study drug to the patient by mail versus making them come in to receive it, wearable devices to capture things like vital signs, electronic capture of patient-reported outcomes, and remote monitoring of the study. And I will say that there's been a lot of creativity with this, in my own institution. In addition to being a regulator, I also practice at Johns Hopkins, and in my own institution, we actually delivered some patients treatment right in their car. We had a drive-up line, and people could get treatment sitting right where they were, so that we didn't have issues of inability to accommodate everyone who needed to get treated based on social distancing requirements, but also to maximize patient safety because they didn't need to be worried about being in the same airspace with other patients at a time that was obviously particularly concerning and dangerous. So just a few things to highlight. FDA's regulatory requirements are the same for trials with decentralized elements as they are for traditional site-based trials. There's no cutting corners. Trials with decentralized elements are welcomed, and I'll show you some data in a moment that these have resulted in regulatory approvals, and they are not all or nothing. You shouldn't think, if I can't do a fully decentralized trial, I'll just do the regular kind. You should say which of these elements would still be able to be incorporated in this trial. And then finally, which of these elements you should be incorporating is really context dependent. It depends on the complexity of administration of that product. It depends upon the patient population that you're talking about. It depends upon the safety of the drug, the stage of drug development, and more. So I think these are the things that you should be considering with decentralized trials. The advantage of them are, many of these are obvious. There's greater trial accessibility and decreased participant burden if we're not bringing patients into a traditional site. This enables enhanced accrual and decreased attrition from the trial as well. And all of this expedites trial completion, which benefits the entire oncology community. In addition, you may have diminished costs. And importantly, I think all of this improves equity and generalizability, which you'll hear this throughout the meeting. Despite a lot of efforts, you will continue to see racial minorities continue to be under-enrolled in clinical trials. They continue to tell us, we want to enroll in clinical trials, but no one talked to me about clinical trials, or there weren't any open at the place where I'm getting treated. So all of this is an effort to improve equity as well as generalizability. But decentralized trials come with some challenges. So there's the issue of reliable access to devices and the internet. How many people in this room either conducted or were a patient in a telehealth visit, at least one during the pandemic? Like everybody? Right? Or your parents did, or someone did, and you all know that the internet goes out, or you can't hear each other, or you can't see each other, whatever. So even in people who may not have socioeconomic reasons to lack access to devices or the internet, there's still issues of reliability. There's patient comfort with technology and the fact that each platform involves a learning curve, and some people may be more comfortable or less with that learning curve. Importantly, there are jurisdictional laws. So are people licensed in the state where they need to be seeing these patients? Or do we have a situation like we had for the first year and a half of the pandemic where you could see patients across state lines as long as you were licensed in some US state? If you're going to use local healthcare providers, you need to train them. And if you're going to use remote DCT personnel, you're going to need to supervise them. And then finally, we have this issue of heterogeneity of data sources. So it's easier to be confident in your quality control if you know that everyone's getting their scans at a small number of outstanding academic centers that have outstanding radiologists. It becomes much more complex when you have, for example, data being collected from multiple different sources. And finally, as with anything electronic, there are concerns for patient privacy and data protection, and these have only accelerated, I would say, in the last couple of years, as I have received, and I'm sure all of you have received, notifications that yet again, some form of your data has been subject to a breach and you are now offered free credit monitoring. Right? It's everybody. I have about 10 of those offers. I'm just going to do them all in succession. So what are the DCT elements that we've seen utilized by sites in oncology trials that have led to FDA approval? So this slide is data from my colleague Timil Patel. This has been submitted and is in press but not yet published, so thanks to him for letting me show it. But as you see, the darker purple there are elements that have been used in oncology clinical trials that have supported approval. And you can see that quite a few of these have been used. So remote site monitoring, remote clinic follow-up or telehealth, remote laboratory assessments or off-site local imaging assessments, remote distribution of investigational product, mailing study drug to people, remote collection of patient-reported outcomes, and electronic informed consent. All of these elements have been used in trials that have subsequently resulted in FDA approval. When we had to abruptly make this decision about how we were going to handle conduct of ongoing clinical trials 2 weeks, not even 2 weeks into the pandemic, one thing we realized was very important is that we needed to collect information on what things were being done in a decentralized fashion. We needed a flag to be able to identify that data so that we could see if data from certain patients or certain types of data from patients turned out to be poor quality. We wanted to know if we could rely on that for that trial in itself, but also broadly in the future. And what we learned is that we can rely on these data. So finally, I'll just conclude with a funny personal story about advocacy-informed trial design. This is Designing Patient-Friendly Trials, a true story. So this was our Bring Your Child to Work Day. This is our beautiful FDA campus, no filter. It's amazing. The kids are all out there playing sports and going to food trucks. And one of the things we have in this incredible day-long day is the opportunity for the kids of employees to enroll in a clinical trial. So these are 5 of the 7 children that my office mate and I have between us. And I'm going to have you just quickly memorize what they're wearing here. People are holding their photographs from previous school years to show how much they've grown since their last visit for this, okay? So the trial that they enrolled in, they're allowed to walk into the front of a room. There's this little side door off to the side where you can exit at the end. So you walk into the front of the room, they don't know what the clinical trial is going to be, and they're invited to enroll in this study where they will have their baseline happiness measured, and then they will be randomly assigned to receive red Skittles or green Skittles. It's a true story. Red Skittles or green Skittles, and then they will have their happiness measured again after eating those. So what's different and why? So you might notice, the blonde is mine, you might notice that she's in totally different clothes and wearing glasses that she does not own. Everybody's changed clothes. The one on the far right, also my daughter in that original picture, has put her hair back and she's put on someone else's jacket. Everyone changed clothes. They ditched the little one in the bathroom because they thought he would be most memorable. Why did they do this? Because they walked in and 4 out of 5 of them were coincidentally randomized to green Skittles, which apparently all children know are inferior to red. Having experienced this injustice, they all left through the side door, went to the bathroom, changed clothes, and ditched the smallest one. He's so cute. Ditched the smallest one and re-enrolled in the trial. This is a true story. This is a commemorative sculpture of me as a regulator that day. I died a little death on Bring Your Child to Work Day. I did not teach them that this was even possible, and my roommate claims that she did not either. All right, so why am I telling you that story? You need to consult patient advocates early in protocol development. And these are the questions that I think that you should ask them. And some of these came from me, and some of these came from patient advocates who were nice enough to answer my plea, either publicly or in direct messages on Twitter, about what makes clinical trials patient-friendly and what makes them not. And so what we learned is, would you be eager to join this study? Just this open-ended question is a great way to start. If you would, what barriers would keep you from participating? Are there expenses that you would need reimbursed to be able to join this study? Are there any exclusion criteria that you think are unnecessary, and why? Is the participant burden, whether that's the burden of having to make visits to a center, or the number of visits, or the frequency of visits, the tests, the questionnaires, the procedures, are these things acceptable to you, or do you think this is too much to ask of people with this condition? Importantly, would you be willing to be randomized to the control arm? Because if the answer is no, equipoise has been lost in the advocacy community, and even if you, as the investigator, believe that these 2 treatments are reasonable things to be randomizing people between, if the people with that disease don't agree with you, you are going to struggle to accrue to your trial. What endpoints are meaningful to you in this setting? Do you need overall survival considering the toxicity of this drug or the line of therapy that you're receiving? Or is progression-free survival okay, for example? Is there an option to crossover? If that's present or not present, is that important to you? Would that be a deal breaker as you decide whether to join this study? And what concerns you most about this trial or would lead you to drop out of it? This is such an important question. And then finally, the things that I think we've historically involved patient advocates more with, which is the informed consent, to say, do you understand this? What questions do you have? Is the language respectful? What would you change, if anything, about it? Is it too long? Will people actually read it? Do we need a summary of it? And then finally, what would you do differently if you were designing this study? And I include this to say, you need to be involving advocates when you are designing the study, not once it's all finished. You need to have them at the table with you as an equal partner right from the beginning. I can tell the trials that have had advocate input from the start and the trials that have not. I can tell as a regulator, I can tell as a clinician, and the trials with advocate input are head and shoulders above the ones that don't have it. So all of that information is going to lead you to the best trial design. So the most patient-friendly study, the most patient-friendly design is going to enable your study to be completed efficiently, and it's going to provide you definitive information about the benefits and risks of that treatment in a sufficiently diverse group of participants to be generalizable to the population. That is the goal. And the most appropriate design, as I said before, is going to be context dependent. That feedback that you get from advocates to inform your decision about which of these approaches are most appropriate to answer your research question is invaluable. For example, pragmatic trials, so this is the middle box there, are intended to enroll a population that's relevant to a decision in practice and representative of the patient or the populations or the clinical settings for whom those decisions are relevant. So these are generally trials with more patient-friendly, streamlined procedures. So less data collection. People are not coming in as often. They're not having to get as much blood drawn. They're not having to do as many questionnaires or procedures. And they're really intended to inform decision makers, patients, clinicians, administrators, policymakers, rather than to be informative about the biological mechanism. So these are the wrong thing to do for a new molecular entity where you're still trying to learn about that target and the first drug in the class. These are things for a pathway that's well established, and you're trying to say, for a group of patients, for example, which of these things might be a better approach, okay? Just to give one example. So, all right. So I'll just conclude here with compassionate use requests. So despite our best efforts, not everyone will qualify for a clinical trial, and there are two sites that I want you all to be familiar with as resources. So the first of these is Reagan-Udall. So Reagan-Udall Foundation is FDA's foundation. It's like FNIH, Foundation for NIH. This is sort of like our FDA, our foundation for FDA. So Reagan-Udall Foundation created a beautiful expanded access navigator tool. So I'm using these terms interchangeably, compassionate use or expanded access, but basically a mechanism for patients to access an investigational agent outside of a clinical trial, okay? So I think that the best way for patients to access a clinical trial and the way that moves the field forward is to be in the trial, right? Not to go in a backdoor or a roundabout way to get the drug. However, there will be people who do not live near trial sites and trials that don't have a decentralized option. There will be people who honestly are pretty reasonable candidates for a trial, but maybe they don't quite make it with one of their lab values, for example. These are great people to consider for compassionate use, patients who've exhausted available therapy. So the Reagan-Udall Expanded Access Navigator, you can see the website up there at the top, explains the concept of compassionate use and expanded access, including FDA's role, and for clinicians, their reporting requirements. It provides company contacts to seek investigational agents and their timeline for decision. So if you're a patient or a caregiver or an advocate, this is your site. And if you say, I wonder how we could access X drug for mom, you go there. It'll say, here's the person to call. That's their phone number. That's their email. They give an answer within 2 business days, for example. Okay? This promotes greater patient equity, and it also allows companies to demonstrate compliance with laws mandating public expanded access policies that every company is required to have. They're not all required to provide compassionate use to their drugs, but they're all required to have a policy and to have that policy be publicly facing so that you can find it. And then this is not for patients and advocates. This is intended for clinicians, okay? So clinicians can use Project Facilitate, which is oncology specific. The previous site I gave you was FDA-wide, any type of drug. This is oncology specific. And basically, this enables clinicians to call or email and talk to a person during business hours in real time about a compassionate use application that they'd like to submit. If they don't have a local IRB, say they're in private practice, they're not in an academic center, they can find a local IRB resource. They can find an expanded access contact for a drug or biotech company, and FDA will help them complete the form that clinicians have to do to be able to provide compassionate use to the drug for their patient. So the contact information is there on the bottom, and there's also an after-hours emergency request line phone number on the bottom as well. So in conclusion, the best people to define patient-centric trials are people with the condition. Patient advocates should be meaningfully involved, starting early in drug development, and their input leads us to the optimal trial design. We have shown that we can make access to investigational agents more equitable, get more generalizable results, and expedite successful oncology drug development while prioritizing and maintaining patient safety through use of modernizing eligibility criteria and including trials with decentralized elements in our drug development programs. And finally, as you've heard, despite our best efforts, some patients may still not be able to access clinical trials, but that does not necessarily mean that they should not be able to access an investigational agent. And these two tools that I've included on the previous slides will help you navigate that process. So thank you very much for your attention.
Thank you very much, Dr. Prowell, for your invaluable insights from the FDA. I did not know that green Skittle is inferior to the red one until today. So for my our next speaker, I will introduce you to Amy. Oh.
I just wanted to add a comment. Thank you so much for the presentation because it included everything that I had in my list, except for a couple of things that I wanted to recommend. That on the ACR report for cancer disparities this year when they issued that report, they mentioned that education is critical for patients to participate in clinical trials. So please do not give up on patients that they said, no, I don't understand, I'm not sure. So give them, maybe you can develop a video explaining the clinical trial. You can develop materials for them or talk with their family members. Just don't give up on those underserved communities that they need to participate because education is going to make a difference. And then mostly is the financial needs. Because our communities, usually they do not have transportation. They do not have the same access to the same centers that we all have. So please just make sure that when you are developing that clinical trial, you include all these issues just to make sure that everybody has the same chance to participate.
I'll just say one thing in response very quickly, which is that FDA put out a statement now about 7 years ago clarifying that we do not consider it inducement to reimburse patient expenses. So people get on a plane, they have to have a hotel, they took an Uber, whatever, any direct expense of the patient to participate in a clinical trial, it's not considered inducement, like coercion, basically, to get them into the clinical trial if you reimburse those expenses. So if someone tells you FDA won't let us pay for that, they are either misinformed or they are misinforming you. You can say, Tatiana Prowell, who works at the FDA, said that on a stage, okay? They are either wrong or they're telling you the wrong thing.
So the next speaker, I would like to introduce you to Dr. Amylou Dueck. She's from Mayo Clinics, and she will discuss about novel statistical design, how we can put the patient first.
Oh, all right. Thank you. What button do I press? Green one. All right. So hi, I'm Amylou. I'm a biostatistician, and there's a lot of things I could have talked about today, but I selected maybe some of my favorites to share with you today. And you know, being a clinical trial statistician, I think one of the things I like the most about my job is being able to be creative, to find solutions, to answer the scientific questions, but to really maximize our ability with every single patient we enroll to answer as many questions as possible. And I think that's kind of what I'm going to focus on here today. So here's my disclosures. So I'm going to start in phase one. That's kind of usually where we think about starting. And I tend to bucket the phase 1 designs into 3 different categories. There's like our traditional rule-based designs. You can think of these as you enroll patients, you have kind of set rules. If you see too much toxicity, you go down dose. If you have acceptable toxicity, you go up a dose, and you just kind of continue in that fashion. On the flip side of that is in the bottom bucket here, these are designs that are based on kind of an underlying model. You enroll patients, you plug their data into a model, and then that model tells you where you go next. So you don't actually know where you're going until you actually see the data. And then we have this middle bucket, which is like somewhere in between. You actually have pre-specified rules. The underlying kind of mechanism, though, is still model-based, but they, on the front end, kind of look like these rule-based designs. So I kind of call them hybrid because they're somewhere in between these top and bottom buckets. So I'm going to show you what these look like. If you've ever read a phase 1 trial paper, I'm going to go ahead and guess that it was probably a 3+3 design, because prior to a couple of years ago, 95% of trials were 3+3 designs. So you've probably actually read one of these already. So how this works is you define a series of increasing dose levels. You define a dose-limiting toxicity. Oftentimes people cut and paste this from a prior protocol, which is a terrible idea, but this is what people usually do. And then you start treating groups of patients, usually in cohorts of 3, in increasing dose levels. If you see, again, low enough toxicity rate, you escalate doses. If you see too high of a dose, too high of a toxicity rate, you basically keep going until you hit that too high a level, and then you back off 1 dose, and you declare that your maximum tolerated dose. And then usually you take that dose level and then immediately move on to like a phase 2 study. There are kind of modifications to these types of trials. You might have heard something like an accelerated titration design, where you could start with, say, single patients to escalate a little bit quicker, especially if you have a lot of dose levels to get through. Once you see kind of your first toxicity, then you kind of slow it down a bit and move into your cohorts of 3. So again, this is very traditional. So the pros of this design: it's simple to implement, low risk of serious toxicity. This, again, widely used, so IRBs accept this, very popular. The negative parts of this trial design is that it's very slow, it's very inflexible. Statistically, these are actually really terrible designs. And even though we know this, people still use them over and over and over again. So this kind of led to these alternative methods that actually use our data in a better way. So in that bottom bucket, these model-based designs, probably the most popular designs are called continual reassessment methods. And how these work is you again, enroll patients in groups. But again, once you see their data, you plug it into a model, and then that model tells you what dose to go to next. So this includes potentially skipping dose levels, but you really zoom in on the optimal dose level much more quickly, and you can really kind of target that optimal dose level in a better way than a 3+3 design. These are just 2 examples of actual trials that were carried out. And I really like this Wheeler publication. I call it my how-to guide. So if you're interested in kind of learning more about it, I recommend that as a read. So the pros here: you treat more patients close to that maximum tolerated dose. These are far more flexible designs. It really capitalizes on all the data. The downside is that these are really logistically challenging to carry out. They're kind of like a black box. Like I said, you don't know where you're going until you get the data. You have to have great communication between your PI, your statisticians, your data folks, just everybody involved in the system. And then you really need to put a bunch of time in the design of these and understand how they're going to work under a whole slew of possibilities. So like I said, I like this middle bucket, which I consider kind of hybrid designs between like these fully model-based designs and these kind of archaic 3+3 designs. My favorite are called Bayesian optimal interval designs, or BOINs. So these kind of look like the 3+3 on the front end and operationally, but on the back end, they actually have much nicer properties statistically because they really are based on a model. And you can see this is kind of a schematic of how an actual trial is carried out. Like I said, it looks very much like a 3+3, but it's actually quite a bit more flexible than a 3+3. So if you're interested in actually playing around with this is a really nice website where you can design BOIN designs to your heart's content. There's lots of ways to modify it. You can actually use a multitude of different toxicity endpoints. You can factor in a biologically optimal dose into the design. You can actually kind of optimize dose selection across various factors. They have a nice decision tree to kind of help guide you to a particular dose level. Just showing here again for your own reference, this is what some of the outputs look like when you actually use this software. So I leave it here so you guys can look at it later. But pros and cons of the Boyne design: easy to run, so it kind of feels like a 3+3, but you're actually doing something quite a bit fancier. It's very predictable, but it still capitalizes on all the data in a better way than the 3+3 design does. The cons: it does take slightly more patience than the 3+3. You also have to kind of put in a few kind of checks and balances to make sure that you're staying on track, which is a little bit different than the 3+3. So if you went to the FDA session just prior to this, you might have heard quite a bit of discussion about a need for better dose optimization. So both of those examples were phase 3 trials that led to drug approvals, and they were still questioning whether that was the right dose. So there's been a movement through Project Optimus to really try to enhance earlier looks at trying to optimize dose. And I think this website is really great, sharing a lot of the work that has been done to try to modify the culture in phase 1 trials. And a lot of it is around, instead of just focusing on the maximum tolerated dose, really trying to go after the minimally effective dose. And also, instead of just picking one dose, potentially considering multiple doses and doing kind of expanded cohorts, potentially in a randomized fashion, so that we get more data earlier. So we're not just immediately moving a single dose and then just kind of pushing it through into like these big giant mega phase 3 trials right off the bat. So this is just a table that kind of summarizes some of these findings. Considering multiple biomarkers or other biological endpoints, you can begin to look at efficacy, but still considering tolerability, dosing schedules earlier in that process, doing an expanded look, considering multiple dose levels. These are all things that we're trying to change culture in the phase 1 setting. All right, so I'm going to skip now to thinking about randomization. So this can come in as early as phase 1 now. So as I said, we're now thinking about taking multiple dose levels and potentially randomizing patients. Typically, we do 1-to-1 randomization. So this means you have an equal chance of getting the experimental treatment versus the standard treatment, say in a typical phase 3 trial. There might be benefits to actually doing an unequal randomization, so say 2-to-1 randomization, meaning that you have twice the likelihood of getting the experimental treatment compared to the standard treatment. So why might you do this? So this gives, if patients are very excited about the experimental treatment, it actually gives them double the likelihood that they're going to get on that trial. So they might be more motivated to participate in your research. Also, say in a phase 2 setting, you might already know a lot about the standard of care treatment, and so you'd rather invest in trying to get more data for the experimental drug that you don't actually know as much about. So it does actually help from you want to invest your time and energy and patience and money in what you don't know, and that's where this can actually help. The major negative that is typically cited here is that these are statistically slightly less efficient. So it is overall generally more patients. And I'll put a little asterisk there that in some scenarios, if you have unequal variances in your arms, that may not be a true statement, but generally speaking, that's a true statement. All right, so I'm gonna switch gears a little bit. If you've ever read a clinical trial paper, you've probably seen a table just like this. So we collect adverse event data in our trials, and it's typically collected using CTCAE criteria. And then it's typically reported just as a maximum grade, summarized as like the proportion of patients that have any grade, and then proportion of patients that have high grade adverse events. Well, this has led to a proliferation of findings in clinical trials that toxicities were well managed, the regimen was well tolerated. I don't think I've ever read a paper where it said that drugs weren't well tolerated. Like that's just that's like a default conclusion in every paper I've ever read. But that's not necessarily the case. These drugs have toxicities. Like I've seen papers, I think, with grade 4 rates of 50% or something crazy, and they still say it was well tolerated. That's a terrible toxicity rate. So there's been a lot of work in recent years to really better define what tolerability means and actually improve how we measure it in our trials. So this was a nice paper by the Friends of Cancer Research that defined tolerability as the degree to which symptomatic and non-symptomatic adverse events associated with the product's administration affect the ability or desire of the patient to adhere to the dose or intensity of therapy. So the FDA has recently come out with their final guidance on core patient-reported outcomes in cancer clinical trials. And a key component of this are PROs meant to enhance measurement of tolerability in our trials. So I'm going to talk about how we do that. So the NCI invested in developing a patient-reported outcome version of the CTCAE. So this is really meant to complement the clinical grading of adverse events. And this is an item library, so it's not meant to be administered all 124 items. It's meant to really select the adverse events that will be applicable for your given trial. So this is what the items look like. This is just for one particular adverse event. You can see the clinical grading here for arthralgia. Very clinical. I don't know what half these words mean. But you can see the patient terms below. We've converted it into patient-friendly language. So arthralgia is now aching joints, such as elbows, knees, and shoulders. And then we ask patients to rate the severity, the frequency, and then the impact on their daily activities. So NCI has the entire ProCTCA library on their website. It is free to use. They also have a custom form builder. As I said, you can pick and choose the adverse events that apply in your given situation. This is just an example of what that form builder creates. And it is available in, I think, 60 languages. So again, really want to encourage inclusivity in your trials, so definitely offer it in multiple languages. The other item that is highly recommended to be included based on the FDA draft guidance is an overall side effect bother item. One question, so it keeps patient burden at a minimum. This is the one that is kind of most commonly being used right now. It's from the FACET or FACT family of questionnaires. It's just a single item that asks, I am bothered by side effects of treatment. NCI also funded through the Moonshot mechanism, a large consortium to kind of enhance the analyses related to tolerability in our cancer treatment trials. This is their website. They just kind of wrapped up their cycle of funding. So kind of all the proposed analyses are all available on this website, so I do recommend checking that out. And it's exciting to see, again, if you were in that session, one of the labels actually included ProCTCAE and GP5 data in it. So it's very exciting to actually see that there has been uptake and changes in how tolerability is being captured, reported, and now actually appearing in an FDA label. So with patient-reported outcomes, I think they are enhancing the information that's available to describe the patient experience in our trials, but it's also important to try to minimize patient burden. I'm a big advocate of using this kind of item library approach to create custom forms. So again, no longer doing the cutting and pasting. Don't just grab tools that may have a bunch of irrelevant items for your patient population. So really using the most modern approaches to make sure that you're keeping your items to a minimum. You can also do fancy things like not necessarily administer all questions at all time points. So direct side effects of therapies, those might change very rapidly at the start of a study, so you might want to measure those symptoms rather frequently at the start of a study. But things like physical function don't change as rapidly. So you do not need to measure, say, physical function as frequently, and so you can spread that out a little kind of less frequently. I'm an advocate of using electronic PROs. However, I'll talk in a minute about making sure that you do offer patients a variety of options in terms of completing PROs. Again, making sure you understand how patients prefer to answer questions and giving them those options. Other advice in terms of PROs, we do have a large kind of clearinghouse for all information available for optimizing use of PROs in clinical trials called Proteus. They have information about how to write your protocol in terms of including PROs, how to select the right PRO measures, how to do proper statistical analysis and graphical reporting of your PRO data, and then kind of how to explain the score changes that you're seeing, and all of that is, again, available on the Proteus website. And then, just capitalizing on the prior comments, really important to talk to patients when you're designing your PRO measurement strategy and how you're going to actually implement this in your protocol. And even right down to how are you actually giving these questionnaires to patients? So we've gotten over the years a lot of feedback in terms of when we design our e-pros, things like, this I think is just common in humans. Who can't stand passwords that are 16 characters long with 14 requirements, and I can't remember any of it. I just tried to get into the speaker ready room and I just about killed their whole system. Terrible. So don't put that in your system. Make it easy for patients to get to their questionnaires. And like I said, I always advocate for making sure you use paper, e-pros, telephone, a variety of options so that patients have the level of comfort that they desire when they complete these questionnaires. All right, 2 more quick topics and then I'm going to wrap it up. So, it's always kind of a question in our adjuvant trials or even our first-line trials, should you allow crossover or not? And we see over and over and over again, a lot of trials do not allow crossover. So crossover is this idea that you dictate the experimental or standard of care options for that initial treatment. And then once the patient experiences progression, after that, sometimes they'll say, you cannot get the experimental treatment if you're on the standard of care arm or some language like that. Whereas some trials will allow patients who are on standard of care to cross over and receive the experimental treatment arm. And so I would say on the kind of pro side, from an ethical perspective, you really want to make sure patients receive the best standard of care possible, because that's your comparator arm. So there is some arguments out there that you should really mandate crossover if that drug has been approved, or at least another drug in that class has been approved as an effective later line of therapy in the same disease. However, if this is literally your first test of the drug in the frontline setting, then you should not allow crossovers. So there is some guidance in this area. So I thought this was an interesting example that I found in one particular paper talking about the CDK4/6 inhibitors, which we also heard about in the session this morning from the FDA. But palbociclib was approved at the time when the first, or when the frontline Mona Lisa and Monarch 3 trials were happening in frontline. They prohibited crossover. So basically, their trials initially were essentially CDK4 inhibition versus nothing. So like even the patients in the control arm were not receiving any CDK4/6 inhibition. So is that really a realistic comparator arm? Well, they ultimately had to go on and do a secondary study to look at whether first line or second line CDK4/6 inhibition made sense. And it turns out whether you get it first line or second line, you have kind of comparable outcomes. So again, it's thinking about they could have done this all up front had they allowed crossover in those initial studies. So there are statistical issues there. If you allow crossover, it does impact those downstream outcomes. But again, sometimes you can maximize the number of questions you can ask, and obviously you have to keep the ethical concerns at the forefront as well. All right, last topic, external control arms. So like I said, I really like the challenge of trying to optimize the data that we use. There are many settings where it may be unethical to randomize, it may be infeasible to randomize. There also might be situations where there might be data available that we would like to use as an external control arm. The FDA actually recently released draft guidance on actually using external control arms, which actually made me very excited to think that the FDA might be opening the door a crack to considering such data in approvals. But I think even outside the regulatory space, I think there are plenty of opportunities to actually capitalize on external controlled arms to maximize our use of existing data. So what I'm talking about here is essentially using potentially multiple sources of patient-level data. This could be from clinical trials, registries, natural history studies, electronic health records, medical claim data, to use as like a control. So then you could either do a single-arm study and then do a comparison with existing data. The downside is that when you randomize patients, you ensure that these are the same patient populations, you have control of baseline characteristics, confounding bias, all of these issues. When you lose that randomization, you do have to be very careful with confounding bias, missing data issues. But these are all addressable, and there's a lot of literature out there that can help with that. And I do think that the guidance that was just released by the FDA actually helps walk through some of those issues. So I'd point you to that if you're interested in these topics. All right, so some concluding thoughts. Many options available in all phases of clinical trial design to enhance the patient experience, both in terms of design, randomization, the measurements you choose, the timing of those measurements, monitoring, analyses, you name it, there's things that can be tweaked. You definitely need to work with a statistician on all of these, so make friends with your neighborhood statistician. And absolutely, you need to work with your patient research partners. You've heard it from Tatiana. I'll just repeat it here now. And then there's plenty of guidance documents and recommendations in the literature. None of this is new, so please use everything at your disposal to optimize what you're doing in your trials. So I'll stop there. Thank you.
Thank you very much for that presentation. I just have one comment. regarding PROs. For patients, when they receive treatment, they do not remember what happened on day 1, on day 2, on day 3. So please try to make it easier for them to capture that and to record it on a daily basis. Maybe you can give them an agenda, you can give them something printed out, or a reminder, a text message, something that reminds them to record how they are feeling. Because if not, it's not going to be accurate. If I come a week after my treatment, I may not remember what happened. So that is the only recommendation that I have for that. Thank you.
Thank you, Dr. Dueck. Thank you, Debbie, and thank you, Dr. Dueck, for the provocative talks about the statistical design. So the last speakers would be Dr. Edith Perez, who require no introduction. So she will talk about collaborations in clinical trials.
Thank you, Sarah. A true pleasure to be here in San Antonio, sharing time with these esteemed colleagues and with all of you. So my topic is collaboration, and I think this topic really fits in very well to conclude the formal comments this afternoon and then open up this discussion time, as I will discuss some of the topics that were addressed actually in the context of collaboration. And if I can advance this one, okay, great. First of all, we really need to be very clear about this, that oncology clinical trials are really the pillars of translating science for benefit of patients. This is the only way we can continue to make advances, conducting the appropriate clinical trials. However, clinical trials are part of a very intricate framework dedicated to the development of new treatments and medications, but they have many steps, many phases, many people who need to know what they're doing, and they need to collaborate to put their knowledge together so that they can be success for the seamless collaboration. And it's not only between healthcare and patients, but also regulatory bodies. In this first slide, I really had to put this in. Patients deserve more effective and safer treatments. We can say a lot about the improvements that have been made over the last few years, but breast cancer remains a huge problem individually, as well as for society. And sadly to say, that the statistics that have been projected by the World Health Organization say, in the world, 20 million persons diagnosed with cancer in 2022, and that's expected to be 35.4 million in 2050. If we think about the number of people who died of cancer in 2022, that was about 8.3 million people, and that number is expected to go up to 18 million. So time is of an essence, and collaboration to conduct the best clinical trials is critical to address this time issue. Certainly, this is also very important because we need to harness the massive amount of improved understanding of fundamental cancer biology, as we need to utilize that information to optimize not only systemic therapies, but also local therapies. We need to think not only about targeting abnormalities of the tumor, but also the immune system. And we need to continue to do our work to personalize therapy, including circulating and tissue biomarkers. And when we talk about biomarkers, it's not only about the biomarkers at the time a tumor is diagnosed, but it's what happens with biomarkers while patients are receiving therapy, after therapy, because that information can help inform, first of all, the understanding of prognosis, as well as novel therapeutic decisions to ultimately improve patient care. The benefits of collaboration are pretty straightforward to me. Number one, enhanced resource sharing. There is not a single person who can do everything, so it's best if we can divide and conquer. And that includes not only finances, but also infrastructure and facilities. Certainly, the other benefit, accelerated trial timelines, as well as enhanced adoption of generated data. And I don't know how many of you thought about this, but a lot of time is spent to collaborate, to develop great clinical trials, and to get the data out, but then the therapies are not utilized because there's a need for continued education, and there's the continued need for all of the stakeholders to be part of the development process. So at the time that the results of the clinical trial are generated, we don't have to try to go back and say, oh my gosh, we didn't include this group as we're developing. So we waste time. where instead of wasting time, what we want is that we want the new information to be applicable and utilized for the patients who need that new information. So, as a clinician investigator, as a patient, as a human being, as a regulator, you have to think of, okay, collaboration is good. How do I work people to collaborate with moi? Okay? And there's some principles here. First of all, if you develop a clinical trial, think of the reason for the clinical trial to be done. What is the expected overall impact if this trial is conducted? And the impact has to be, I think, addressing patients, but also there may be some trials that are heavily involved in trying to understand biology and science that can ultimately lead to other studies that will help optimize patient care. We need to get funding, because we can have the greatest idea, but if we don't have money to do the clinical trial, that's a problem. So if you as an investigator or as someone who's thinking of a trial can conceive not only how to collaborate to develop the protocol document, but how are you going to collaborate to get funding? So I tell you, it pays off a lot if you go to a colleague. If I go to anyone, my colleagues at Mayo, and I said, I already got $10 million, do you want to be part of this? Instead of saying, well, the idea is good, it may take 5 years to get some money. So think about how to optimize funding, again, in very collaborative fashion to be able to then get more people to want to work with you. Additional criteria that are utilized for people who may consider collaborating with you include, how long will this take? Is there adequate protection for human subjects? Is there equitable inclusion of individuals? What are the milestones for the study? What is the statistical plan that has been outlined that will help determine whether the data that are generated are leading to optimal results to make the future decisions? The other aspect is, when we develop a clinical trial, we have to think with our collaborators, not only to start clinical trial, but how are we going to sustain the conduct of the clinical trials? When we run a clinical trial, let's anticipate challenges that may occur ahead of time, so that we can properly and quickly address them, because the bottom line is we want the clinical trials to be completed, for the data to be generated. And last but not least, again, having an appropriate data sharing plan is extremely important. So, what types of collaborators can research teams consider? Certainly, we can do academic and private clinical practice institutions. Locally in the United States, there are many NCI-sponsored and other translational research centers. Specifically, as of a couple of days ago, there were 57 NCI comprehensive cancer centers, 8 clinical cancer centers, 7 basic laboratories cancer centers that can be utilized for the conduct of trials. However, one must also remember that approximately 80 to 85% of patients who are diagnosed with cancer are treated in the community. So we must think about optimizing collaboration between academic institutions and private hematologists, oncologists to optimize data generation. Another important collaborator is biopharma. Pharma is not an enemy here. Pharma develops agents with the idea that they will improve people's lives so that the shareholders also have some benefit. It's really a very important issue, and I hope that as we all advance in our careers, if we hear someone talking about the dark side, when we talk about biopharma, we stop them right that moment. We have to understand that everybody has a potential role, and if we work together, the benefit for patients will be optimized. You need to think about regulatory bodies, the FDA, the EMEA, patient advocacy groups and foundations, medical education groups to really disseminate the data. This is a consistent type of slide, as you've heard from colleagues today. Engaging patients in collaboration just makes sense. And always, always consider that involvement early. And it's not only for the clinical relevance. I think we need to include advocates also when we have the scientific discussions related to biological rationale. Certainly logistics a big issue. How many visits, frequency, and the burden of diagnostic tests and biopsies. And I tell you, I've been in many meetings with persons who were developing a trial, and I can't tell you how many times they want to get biopsies on patients. And then I call that into question, or somebody calls this into question, and I said, well, we need to understand what's happening. And I said, but what is the impact on the human being who has volunteered to participate in the clinical trial? Do you absolutely need biopsies every month? And sometimes, people don't actually think about that aspect. So we can bring that perspective and listening to the patients related to the potential burden in the context of the clinical trial being considered is extremely important. But to me, I put something a little extra on this slide, which is, it's more than the biopsies, it's how long it takes to get to the clinics. Who's going to pay for parking? Who's going to pay for the meals? Who's going to pay, deal with the time away from family, work, and friends? Those are factors that are critical when we think about optimization of the ecosystem for clinical trials. And certainly information related to the perception of the informed consent has already been discussed. In the next few slides, I'm going to talk about a couple more ideas related to collaboration, and then I'm going to talk about what's novel, what's new, what's different related to the collaborations that we were doing a few years ago compared to what's possible now. Certainly, we can do multi-center trials. We can do trials, between consortiums and networks. But one of the areas of collaboration that is gathering a lot of interest is what we call the public-private partnerships. So these are joint projects between government and industry. And I had an opportunity to participate in a National Academies workshop in October, where we had actually a full 2 days discussing the rationale for public and private partnerships and making recommendations of how they can be optimized. And the report just came out in just last week officially in the National Academy's website. When we think about these public and private partnerships, again, it brings people together. It's not that the public arena does things. It's not that the private or biopharma does another thing. Must work together. What are the challenges, though? We have all these novel ideas, grandiose ideas, but sometimes there's some challenges that we need to think about, and they may include misaligned goals and expectations. So we need upfront agreements to be set up. And I tell you how many of you are enthusiastic about addressing challenges. Sometimes what we do, we see a challenge and we don't even want to talk about it. We just kind of keep it to ourselves and hopefully things will get better. To be able to truly collaborate in the context of trials and research, we must address issues as early as they are identified. Certainly, we need to deal with data privacy concerns, and they may be different for different people. There may be logistical hurdles that need to be addressed. And the other issue for investigators, and several of you are writing protocols. I can tell you there's a big difference between now and the way it used to be, and Amylou, you remember this. When I was fortunate to start my career, kind of starting my career, as a PI, I could actually go to somebody. I said, I'm going to do this trial. I would get a colleague statistician or the colleagues at the clinic and say, we're going to get this done. I would go to pharma or to the NCI and try to get support. Now, as a principal investigator, you have an idea, and you have to go to 5 committees in your own institution, to get this done. And that is not that nice. It actually slows things a lot. You guys are smiling, because you see this happening. The issue of control of who's going to drive this is really very important. And then prioritization. You can have a great idea, but if other people don't see that that's also their priority, it's going to be hard for them to collaborate with you in an effective way. So best practices, we have the ideas, we have some challenges. How are we going to do this? Inclusivity and respect between all stakeholders. There's no one that is better than anybody else. I always think about the knowledge that we've all acquired. And when we were born, none of us knew many things, and we learned. So I really think that people have the ability to learn if they want to, and we need to respect each other. We need to have the defined shared objectives. We need the governance workshop and clear communication channels for all stakeholders on an ongoing basis. There are many evolving trends in this space. There are more global trials. There are more academic and community-based trials. There's more intent in equitable research. You know, Tatiana, you talked a lot in a very nice way about the importance of the centralized trial, the importance of patient-reported outcomes. We're going to talk a little bit about real-world data, more communication tools, more data sharing platforms, cloud-based systems for ongoing analysis of data, and last but not least, artificial intelligence. So let's talk a little bit about artificial intelligence as I have only about 5 more slides this afternoon. Artificial intelligence can truly enhance many health solutions, including clinical trials. You know, as I look at this for the potential role of artificial intelligence for consumers and patients, for medical providers, for biopharma, for payers, in all of these buckets, clinical trials can be considered. Artificial intelligence is also allowing us to essentially harness the changing evidence landscape for data generation. You know, traditionally, on the left side of this slide, we do phase 1, 2, 3, some phase 4 clinical trials, and then we get the data generated. And what's happening, though, is that we know the majority of patients are treated with the drug after the drug is approved. So after the phase 1, 2, 3, or even some phase 4 trials are completed. So there are a lot of data that are out there, and we need to harness the potential to use artificial intelligence to get that information of what is really happening to individuals who are receiving this treatment, so that then we can take that information, bring it back to determine the best patients to receive a therapy or modifications that should be addressed in future trials. This slide is busy, and the next slide is busy, but you'll have these slides, and you can just hopefully use this as a helpful system for you. But there are many uses of artificial intelligence for drug development, drug target identification, selection, prioritization, screening and designing compounds, modeling pharmacokinetics, advanced pharmaceutical manufacturing, and certainly clinical trials. And you can see some specifics related to these specific areas that I will not read this afternoon. But to make things even more complicated and hopefully clearer at the same time, because you'll have these slides, I think of any of these buckets, what are specific examples of how artificial intelligence can be used in a collaborative way as opportunities for investigators and patients to really get the work done. You know, and I'm going to just take the patient recruitment one. You know, you can mine data from databases, trial announcements, social media, the literature, to figure out how to enhance patient recruitment. You can think about adherence, and retention of patients in clinical trials. Think about this. You can use innovative methods to communicate. If people don't come to a laboratory, you can send them a little email, or they can go to the web, et cetera, et cetera. So they're tangible, well-described items that we can harness utilizing artificial intelligence to optimize the ability to just take information from many places to do what we need to do. And then this is the last data slide, because I think that in order for us to really make it the difference that we want for patients, we need to connect diverse data sets across patient populations. And that not only includes the data from the phase 1, phase 2, 3, 4 clinical trials, but look at utilization records, look at pathology, look at pharmacy claims, medical imaging, a lot of information, all in a collaborative way. So as a conclusion, collaboration holds the potential to accelerate medical research, enhance patient care, and drive innovation in healthcare delivery. By harnessing the power of relevant integrated data, research teams can unlock insights, improve outcomes, and advance the understanding and treatment of diseases for the benefit of patients and society as a whole. Call to action for stakeholders, all of us. Encourage cross-sector partnerships. Carefully analyze all accumulated data and put the data together. Stay abreast of emerging technologies and industry, because then we can take advantage of these advances to optimize patient care. Let's innovate. Thank you very much. And the references for many of my slides are here, as you see, including ChatGPT. So it was just, I'm doing this these days. I have a topic, and I'm actually going to one of the sites. How many of you are doing this? Okay, all of the speakers, several of you. That's taking advantage of what's new so that we can do our job better. What I found out, right now, it's great if I'm going to give a lecture to a high school student. It's great to use ChatGPT, but then you can build from there. So I'm just very excited about the potential, guys. And I'm very appreciative of your time this afternoon. Thank you very much.
So I know our time is short. We had fantastic content, and I'm really glad that we were able to deliver a lot of the topic that was discussed. I don't know that my voice is coming through. There we are. But in an effort to also respect your interest in other sessions that will be coming this evening, I don't think we'll have as much of an opportunity for questions. I did want to give a little bit for the conversation in the room, and there was one question that came through online, so I'll just mention that at first, and then we'll take it over to the questions in the room, and then we'll allow you to get on with the rest of your evening. The person from the online audience or that had submitted a question was really about how patient advocates that may not be in formal constructs can get involved in the conversation with clinical trialists. And so I'll open that up, I think, for the panel, but maybe for the patient advocate first, as I know that you've been successful in navigating that as well.
For me, it's very important that we start participating since the beginning. Sometimes I get a request at the last minute saying, I need a letter of support for this research that the deadline is next week. And I said, unfortunately, I will not be able to write that letter of support because I don't know anything about your research study. So really, this is where we have to value the participation of the patients since the beginning, because we will support you, but if we have knowledge about your research.
Thank you. I think that's really what was said as well, is that please keep in mind the respect for the patients, that it takes some time to really give you an adequate review. So for the researchers in here, I think that that'll be an important thing that hopefully stays with you. And to the microphone on the left here, please introduce yourself.
I'm not sure it's like Marisa Weiss, breastcancer.org. Two things. One is in these collaborations, there's often a conversation about who owns the data. And I think it's really important that the patient own her own data. So that's we do original research at breastcancer.org, but that is always a tenet, the patient owns her own data, and that builds trust. The other thing that's important, I think, to think about is that, as Dr. Perez has said about the importance of care in the community, 85% of care is provided in the community. Well, there are digital communities like at breastcancer.org where all these people are coming for their care, and in terms of patient-reported outcomes and real-time capture of sentiment as well as adverse events and all that, you can design a community online where people consume things today by connecting with other people through community. So I would like to, throw that out there, is that perceive that as a community of care, not just a hospital setting and outside of an academic center, because you can reach more people that way. They can have their source of support and data sharing that way. And you can do the decentralized clinical trial data that way, those ways as well. Thank you.
Thank you. I think that's really pertinent, and I think that as we look to data sharing and also just crowdsourcing how people live in the real world, that becomes really meaningful as we think about design and obtaining that information. So thank you.
I wanted just to add that some organizations, non-profits, they are building databases with patient information. And we want to do that because that is very important to share it with different organizations so they can do research a little faster. So if you want that information, we can share that information for you to recruit more patients, share that information so it's available for many researchers.
I'll just add one other thing, which is that I've actually been a participant in 4 trials myself as a healthy volunteer, because I feel like it makes you a better trialist and a better regulator if you've been a participant. And one thing that I personally have appreciated a lot is when the investigators send you the results of the study at the end. So the most recent one I was in was attempting to get multiple different SARS-CoV-2 tests approved early in the pandemic. And so I was in this study where I had to do swabs for all these different tests and submit them. And I mean, it was a lot of work. Like I was swabbing myself endlessly, driving these things to FedEx every day. It was really a chore. And I appreciated at the end when they sent me a letter and said, thanks for your contribution. You were one of this many patients, and these drugs, actually, these tests actually ended up getting approved by FDA as a result of this study. And I appreciated that because it really was a time burden at a time as a clinician during a pandemic, I didn't have a ton of time. So just think about that if you're an investigator. Give that feedback to patients of how the study turned out.
Yeah, I think that's really important to honor the contribution.
Hi, I'm Jade Baran. I'm a patient advocate, and I had a comment about the question online about patient advocates, how they can get involved. And I actually am going to hand that back to you guys because it needs to be a requirement. It needs to be in the guidelines for the IRBs and say, look, you're not as likely to get this grant funded if you don't put us in there. I feel like that is really important because there won't be the motivation otherwise. So my question was about AI, actually. It's for both the people. In terms of those questionnaires, there's a lot. And I always have a hard time filling them out because getting quantitative data out of the human experience about my feelings during the week is very, very hard. And I would love to see qualitative data, how I'm feeling, interviews being used rather than these little numbers on a slide. And I actually think that AI could be very effective if you could do it face-to-face with a human, but have it recorded and figure out, are these side effects really bad? And also link it to the attitude about it. Because sometimes, I have metastatic breast cancer. As I get closer to the end of my life, if I really want to hang on, I'm going to be willing to put up with a lot more. I still might be incredibly distressed about it, but my motivation and my attitudes about that are going to be different. And I feel like that is something that you could leverage. AI, it would also bring something down, something that is very annoying to patients to have to do and doesn't really get at the things we're experiencing. Thank you very much.
That's a great comment. First, thank you for being here and for your contributions, and hopefully things will work out for the better. Number two, systems already exist where a physician is seeing a patient and the conversation is being captured and then summaries are produced. Actually, I'm doing that for all of my meetings. Actually, it's happening. And 2 minutes later, I get a summary of what were the main topics, and if I want a transcript, all of this is available. So this is widely available now. So there's some positivity here. It's moving in the direction that you nicely outlined.
And I'll maybe just add, there's a lot of work right now to move PROs to earlier phase trials where we actually know less, and one of the recommendations is to use exit surveys and more qualitative methods because we don't actually know what we should even be asking. And then maybe one more point, kind of our largest quality of life meeting is the International Society for Quality of Life Research. Our theme next year is all about AI, and I do think that we are moving in the direction of chatbots and actually leveraging technology. I do think our field of kind of quantitative measurement is rapidly changing. So you're probably a thought leader here in your comments.
Hi, I'm Judy Fitzgerald from Citrus for Prevention, and I want to build on what Debbie said and what the last person said as well. I feel that we're losing a lot of information with the PROs because, again, it's not really individualized, and what Debbie said, I may forget what I felt last week, but if there was interaction with the patient, not only physical but psychological, to tie into what the last person said, and the other thing that I found being an advocate on a trial, so because we have to respect the privacy of the patient, the research people never know what's going on with the patients. And all we know is what the oncologist is sharing with us. So if there was some way that there could be a system where the patient would put in information every day, as Debbie said, a prompt or an app or something, that they could put in what they're feeling that could also be shared with the other patients on the trial. Because let's say I'm on the trial and I get a headache today. Well, is that because of my drug or do I have brain mets? You know, and so I think the patient could really be put more at ease to be in the trial, and they actually, I think, would be more truthful with their side effects, because there's always that case where if a drug is working, I'm not going to say anything because I don't want to get taken off this trial. And so I think if there was some way of better communication and better collection of data, and the other thing also is once the trial is over, nobody says anything about what's happening later in life. So we don't know what the side effects are from that drug 5, 10, 15 years later. And so I just feel like we're not really getting all the information that we could be getting. And the loss of information, again, so going from phase 1 to phase 2, the phase 2 people don't get any of the information from what the phase 1 people told their oncologists. So I don't know how we overcome that, but I just feel like we're really losing a lot of valuable information. Thank you.
Thank you very much for that input. I think it's very meaningful.
Elena Shagi-Saltanova, University of Colorado. I have a question to Tatiana. I'm a medical oncologist. I do clinical trials and translational research, and we talked here about how crossover can impact our analysis of overall survival data. But I can tell my personal bias is sometimes it's kind of difficult to interpret overall survival data when there is no crossover. And a good example will be, for example, like the drug is already approved here in the US and moving to the earlier lines of therapy, something like Destiny Breast 26, right? So we know it's hugely beneficial, everybody gonna get it here, but if you do this study somewhere in Eastern Europe or say Africa where people will not have access to the drug, it's very easy to show overall survival. So I just want to know what's the FDA position on that, or how as clinicians we should actually be interpreting this overall survival data.
Yeah, it's a great and really important question. You know, I will tell you as a regulator that I definitely never want a patient to be denied an effective subsequent therapy. And often if that therapy is available and you don't provide access to it in crossover, people will simply drop out and get it. And so at that point, the outcome is still, you know, impacted, but now you don't know how it's impacted. All you know is the people left your study, right? And so I would prefer to have that outcome be impacted in a way that there's still good control over the data. And we have, you know, even in trials where you would think everyone would cross over, everyone doesn't cross over. Like what we find is that even in patients who've had disease progression in a trial that permits crossover, not everyone will choose to do it. And this is maybe a question where, you know, we need a biostatistician to weigh in here, but there are ways that you can analyze the people who did not cross over versus those who did, or the study prior to crossover being instituted, you know, or initiated versus, you know, after it was initiated. What I will tell you is that if you have a drug that will impact overall survival in any line of therapy, whether it's now or subsequent, and you allow people to cross over to it, you are going to confound that outcome. Like, it's a fact. The question is your drug still effective enough that even if that overall survival benefit were smaller, it would still be clinically meaningful? Even if that overall survival benefit is smaller and a lot of people cross over, will you still achieve statistical significance in that endpoint? And whether that matters depends on where that endpoint is in your statistical analysis plan, right? So if that's your primary endpoint, if your primary endpoint is overall survival, and now it's not statistically significant anymore, you're going to have trouble there because you need to demonstrate, you know, a statistically significant and clinically meaningful improvement in that primary endpoint to get your drug on the market. I think that the more common scenario that we have, because certainly in breast, overall survival is often a co-primary endpoint or it's a subsequent endpoint in a hierarchical analysis plan, but it's not the standalone primary endpoint very often. I think the challenge that we have is that these studies are global, and maybe Edith can weigh in here as someone who's been in industry, but these studies are global, and even if FDA says we are satisfied with PFS, for example, we don't need to see an improvement in overall survival to put this drug on the market. What you may find is that in another region of the world, they may say, we disagree with that. Or they may say, we also will approve it, for example, EMA, but then individual member countries won't be providing it or covering it for their citizens, and therefore the drug is approved, but effectively not used. And that's problematic for industry, but it's also problematic for patients, right? Like I don't want as a regulator, and I struggle with this every day, think about this. Like, to whom do I have the greatest responsibility? Is it the people in the trial? Is it everybody who has this disease currently in the US? Is it everybody who will ever have this disease in the US? Is it the whole global oncology population? Because the fact is, these patients' interests are directly in conflict with one another sometimes with these issues about trial design, like, should crossover be allowed? And at the end of the day, this isn't just like, this is the hardest thing about my job, is this is not theoretical. I'm like, it's not an academic thing where I'm like, yeah, that's tough, you know, because this one group would benefit more from this and this other group would benefit more from that. Like, I actually have to make a decision and tell a company, like, this is what needs to be done in your study. That's the hardest part of being a regulator, is you have to make a binary yes-no decision about approval, about design issues, about whatever. So what I will say is, I think that when overall survival is a secondary endpoint, it's not the key standalone thing that will support regulatory approval. I think that in many cases, a very good case can be made to allow crossover, and you just have to have statistical analysis plan prospectively defined, outlined how you're going to handle that information. I don't know if you want to add anything, but.
I think for the sake of time, this conversation incredibly meaningful and really important, but I know you also have other things that you probably want to see tonight too. Thank you so much for our in-person online audience. I'm really hopeful that with your conversation and engagement, we'll continue to build sessions like this at SABCS. And to our patients, we are incredibly in debt, not only to the input, your advocacy, but also to the ability for people like myself who've made careers out of helping you have greater hope. So thank you for contributing to the research, but also to our lives and to this work. Have a good night, and we hope you enjoy SABCS.